Hi, I'm Mark Moser from Mississippi State University in the Department of Agriculture and Biological Engineering. So, my poster is an initial screening of pomegranate punicallion for the use in intra-articular osteoarthritis therapy. So basically, we're looking at the effects of punicallion on articular cartilage in the joint. Our aim was to perform initial screening. It's been shown in former studies that it has an effect on chondroprotectivity, collagen protectivity, and pretty much immune suppression in the joint when used in oral stimulation to mouth. So we looked at the mechanical behavior, thermostability, and collagenase resistance in it. Also, we did cytotoxicity study about its viability in vitro. So first of all, we took 5 millimeter diameter osteochondral plugs harvested from adult porcine stifle joints. We fixated them with 30% punicallion extracted from pomelo fruit, dissolved it at 1% weight per volume in PBS containing 30% DMSO, and let it orbital mix in 37 degrees for 24 hours. Then we separated from the bone interface. So we did an unconfined compression test to start with. It was stepwise with a 5% strain amplitude. We then measured the peak and equilibrium stresses from each step we plotted against the strain. The slopes were then best fit to each group, and we determined the instantaneous and equilibrium moduluses. And that was for our mechanical properties. For the thermostability, we sent it off for TGA and differential scanning calorimetry services provided by More Analytics. They were freeze-dried with a control and a punicallion-treated disc. For collagenase resistance, we rinsed each piece in distilled water, freeze-dried it, and weighed it. Samples were then incubated for 24 hours in 37 degrees under orbital shaking with 1 ml of 0.1% weight per volume type 2 collagenase. And once they were done, they were washed, refreeze-dried, and weighed. And we compare that controls and punicallion experimental values. Next, to do cell viability, chondrocytes were obtained from the femoral articular cartilage of a Sprague doll rat, and they were expanded in monolayer with DMIM and 10% FPS. And at fourth passage, we see the cells into a 96-well plate. And they were grown to confluence in each of those plates. We then used 98% punicallion extract for this one. The cells were incubated in the solution for about 8 hours, and then washed twice with PBS. And we then estimated the cell numbers using a cell counting kit, and viability was calculated as a percent average of the controls. For the last part of it, we did a live dead staining using the distal femurs of a porcine neonatal stifle joint. Experiments were then exposed to the same 98% punicallion extract for 30 minutes. Then we cut microslices about 500 microns and washed those in PBS 10 times and applied the live dead cell viability stain and then viewed that. So the results of each experiment were punicallion had a good effect on the instantaneous modulus, but didn't really have an effect on equilibrium modulus, but didn't harm it either. It's very close to the controls. For the collagenase resistance, it showed it did have a clear effect on it. I suppose for over the 24 hours, showed about a tenfold increase. So, for our thermostability results, showed it didn't really have an effect on the thermostability of it. And for cytotoxicity, 100% controls, no difference, wasn't cytotoxicity at all. And the live dead staining showed good green bands right here for live cells. These would be punicalin, and this would be the controls, showing basically no cytotoxicity at all. So what this tells us is it does have chondroprotectivity, but since punicalin is a larger molecule, it's affecting most likely just the articular surface. Through the instantaneous modulus, it shows that it's affecting the direct permeability of the tissue, but in a longer stress, it's not affecting that. So it's not affecting the inside of the cartilage tissue and affecting that ECM, which also is shown by the thermostability that it did not increase, no extra temperature was released, no extra energy was released compared to controls. In fact, it was a little bit separate. So this does show it probably worked well in interarticular osteoarthritis therapy in fixing focal joints or focal defects in the joint. And look forward to doing more work with it. We're trying to move on to animal studies now that we've shown all this, and that's what we have up in the future.
All right. Thank you very much.